And then apparently she's been in such severe pain for the last 4 months that she doesn't really eat or drink, and hadn't had anything to eat or drink for 3 days. So she's been in the hospital since yesterday morning. But I feel like she's gonna be okay. Maybe that's my change. But my mom's kind of not so nice about it.
Oh no.
Yeah, it's a lot. It feels.
Okay, so
without like actual.
For what we have left is we have a lecture quiz today, then we have take-home quiz next week. That's probably just due before the exam, I'm assuming.
Yeah, that's the hardest one to get back.
Okay, yeah, I figured. Okay.
By the way, don't put it. You gotta get that
2 lecture exams left, 0 lab quizzes left, 2 exams left.
Oh.
And then
And
semester
one lab.
project.
Lab today, and that's it.
Oh, and this is our last lab day on the schedule. Is that still correct?
Yes.
Okay.
And today's blood typing.
I love blood
Yes.
typing.
It's
Thank God.
simulated. We don't all get to share the same needle.
No, I've seen the simulations.
Try not to do that.
Why?
I know it's much more fun, but
It's much more fun to share needles. Saves money. We're just trying to save the department money.
well, the department saved itself a lot of money by not doing.
It's blood source. Because we've been parking lot space, and we'll help them advertise, and we'll give them like free kazookies and stuff. The Zookies. I was told that for the last year and a half I've been saying the Zookie. Because it's cookie. What's a cookie?
Do you know when we're going to be getting the take-home quiz? Are we getting it next week, or would we get
5 minutes before 2.
Next week, we only have tonight.
So if we got it today, you want it today?
Yeah, because it gives me more time to get it over.
Well, I don't have it, so you get it
Next
Tuesday.
and then it's due the following Tuesday.
Well, it's due before the semester ends. So I don't want them to not turn it in.
Yeah.
That's the hard part, getting it back.
I'd be happy to make copies for you.
I don't have something written.
Oh. Well,
Okay.
let's keep it that way. Have a little mercy. Come on.
Wow.
I'm trying. I never do this.
No, the more points,
That's
the
true.
better.
That's
So, and it's interesting, education is one of the places where people ask for less, for the same price. Can we have less, please?
Well, we're still learning.
I know.
We're just not testing.
Yes.
Is the adaptive and acquired immunity homework assignment?
That is a homework assignment. And so the material that we're going to go over tonight can largely be used to fill that in. So yes, that is an assignment. Adaptive and acquired immunity, it's a homework assignment. Another 20-pointer. Okay, so this was the last slide that I showed you last time, I think, but I'm not sure. Yes. So what we're going to do now is talk about how we can divide adaptive or acquired immunity into 2 categories, and then talk about those categories in greater detail. I think I may have given you the terms humoral immunity, cellular immunity. Okay. So humoral immunity involves B cells and the production of antibodies. Cellular immunity involves T cells and the production of cytokines. So they're a little bit different. They're not entirely separate. We're going to see how they overlap, and also how they overlap the innate immune system. So we like to, in biology, divide things into categories and then talk about how these categories kind of overlap one another. That's just how it is. So humoral immunity. For a long time, people talked about the humor, and what they were talking about was fluid within the body. The blood, the lymph. So humoral immunity has to do with these antibodies that are circulating. Some of them are in the bloodstream, some of them are in the lymphatic system. At any rate, humoral immunity is also called antibody-mediated immunity because it does involve B cells, and B cells produce antibodies. Actually, B cells have antibodies on their surfaces, and they also give rise to cells called plasma cells that secrete antibody, release it into the surrounding environment. So that's one arm of the adaptive or acquired immune system, is humoral immunity. The other is called cellular immunity or cell-mediated immunity because it involves T lymphocytes, and the T lymphocytes don't produce antibodies. For a long time, the chemicals that they produced were called lymphokines, and then it was determined that these chemicals were not strictly produced by lymphocytes, and so they changed their name to cytokines. As far as I know, cytokines are just a group of high molecular weight substances. I'm assuming they're proteins, but then part of me thinks maybe they aren't all proteins because They're just described as high molecular weight substances. Anyway, so we found out a little bit about helper T lymphocytes. Helper T lymphocytes are essential to both cell-mediated immunity and humoral immunity. And so, as I said, there's overlap between these two systems. Well, so in order to understand how the adaptive immune responses are initiated, or at least the way I want you to see how they can be initiated, we need to take a look at some of the players that are involved in this. So we're talking about a system where B cells produce antibodies and release antibodies into the circulation, and antibodies are typically released in response to antigens, some kind of foreign agent getting into the body. Well, so we need to talk about these in a little bit more detail. If you have that homework, the adaptive or acquired immunity homework, there's some things that you can fill in as we go through this section, or not, because you can fill it in later too, take notes. Anyway, one thing I wanted to point out, and I've been mentioning a number of things where spelling is similar, but be careful. So antibody and antibiotic, these are two words that you need to really be careful about. Sometimes people look at the front end of a word and then guess what the word is without really sounding it out. And what's worse is antibody and antibiotic are both often abbreviated capital A, small b. Try to avoid doing that because antibodies and antibiotics are completely different, not related to one another at all. So try to avoid using that acronym, that abbreviation. The terms are not synonymous. Antibodies and antibiotics, very, very different. So what are antibodies? Another term for antibodies is immunoglobulins. As a consequence, you'll see these abbreviated sometimes capital I, small g. Capital I, small g stands for immunoglobulin. They're globular proteins. They're quaternary proteins. So they have more than one polypeptide chain. The biggest ones have way more than one polypeptide chain. Typically they have 4, but they can have more than 4. They're made by lymphocytes. They do occur on the surfaces of lymphocytes, so they're stuck to the surfaces and they have their antigen binding sites kind of extending outward along the outside surface of the cell. But these things can also be released by plasma cells so that they can be in the circulation, in the humor, again, in the blood, in the lymph, as free proteins. There are also antibodies that are typically associated with mucous membranes. I mentioned when talking about mucous membranes that there were antibodies in the isotype IgA, but I didn't talk about what isotypes are, so now we have to go back and talk about that. These diagrammatic representations that you see here where antibodies have a Y shape, these are all representations of gamma globulin or immunoglobulin gamma, which is IgG. So human B cells will produce large quantities of specific antibodies in response to specific types of foreign agents, antigens. As soon as I say foreign, and I think, well, usually these chemicals that get into the body and stimulate antibody production, and the term antigen is kind of like surfactant. It's a shortened word because what it stands for is antibody generator. The idea was that some kind of a chemical would generate antibody production. It's not quite that simple because what the antigens do is cause cells to produce antibody in large quantity. Nevertheless, we think of them as stimulating antibody production. So antigens, antibody generators, most of the time when we're talking about, because this is a microbiology class, these foreign agents that can get into the body are microbes. So they're bacteria, or they're viruses, or they're protozoa, or they are fungi. They can also be toxins, and it gets more complicated than that. So antibodies are produced. They can bind very specifically with antigens, and there are, remember I said there are all these subpopulations of B cells. Different subpopulations of B cells will produce antibodies that have slightly different amino acid sequences in their structure. Consequently, their antigen binding sites are different. When they do bind with antigens, it's not covalent bonding. It's weak bonding so that it can form and release. The bonding between antibody and antigen is very much dependent upon concentration of these two substances. Anyway, so they can either be hydrogen bonding or ionic bonding. They're not covalent. Now, antibodies in general are divided into 5 groups or categories, or we can call them classes. Technically, these are called isotypes. Here's another place where you have to be very careful about spelling, because many people are familiar with the term isotope. And isotope and isotype are not related to one another at all. Isotopes are atoms with different numbers of neutrons, I believe. So we have a different atomic number. Isotypes are different categories of antibodies. Iso means same, so isotype just means same type. Well, there are 5 isotypes. Again, not isotopes. You see them illustrated here with the Ig in front. We have IgG, IgE, IgD, IgM, and IgA. Oftentimes you look at this and think, well, why are they such an odd accumulation of letters? Because a lot of times when things are identified, they're identified as A, B, C, D, E. Well, these were, except the letters that were used were not the English alphabet, they were the Greek alphabet. And so the letters were alpha, beta, delta, gamma, epsilon. But then beta got replaced by mega. And so we don't have a B, we have an A, we have a mega, we have a delta, we have a gamma, we have an epsilon. So that's the sequence of letters that are labeling the isotypes. If it helps, you can remember that they actually spell a word. The IGs, all the letters that come together, they spell MADG, M-A-D-G. Do you really need to know that? Well, you should know that there's 5 different classes or isotypes and what the letters are. So again, they're labeled by the Greek alphabet, and B was replaced by M because there's one type of antibody. The M is for mega, and they're extremely large. So here we see the IgM. These dark points here are the antigen binding sites. So IgM has 10 antigen binding sites. Most of the other antibodies have 2. IgG has 2, IgE has 2, IgD has 2, IgA has 4. So, which ones of these are the most abundant? This is a question that is asked on that homework assignment. In actuality, humans produce mostly IgG and IgA. But the question isn't which ones are produced in greatest quantity. The question is which ones are greatest or in greatest concentration in the circulation, in the blood, in the lymph. And the answer to that is IgG and IgM. The reason it's not IgA, even though IgA is produced in large quantity, is because IgA is associated with mucous membranes. They're actually stuck to mucous membranes. They're also in secretions associated with mucous membranes. So they're in breast milk, they're in tears, they're in saliva, nasal secretions, that kind of thing. But they're not in the circulation in large quantities. So of the antibodies that are most important to us when we start talking about what antibodies do to defend us, IgG and IgM are the most important. IgA, as I said, are associated with mucous membranes. And then IgE, we're going to see, are associated with hypersensitivity reactions. We're going to talk about hypersensitivity tonight. Hypersensitivity reactions are allergic reactions, and type 1 hypersensitivity involves IgE. There's some other types of hypersensitivity that involve other antibodies. For a long time, there was no mention of what IgD was for. Now, apparently, they've determined that IgD has a regulatory function. So you're having some kind of activity as an interface between inflammation and the immune response. Don't worry, I'm not going to ask you questions about that one. Now, this shows you the structure of what's called an antibody monomer. That means you've only got one antibody, but notice it's a quaternary protein, so you've got 4 polypeptide chains. These polypeptides are long, 2 of them are long, or these are referred to as heavy chains, and 2 of them are short. The short ones are referred to as light chains. These polypeptides, these amino acid sequences, have a constant region and a variable region. In this illustration, the portion of each chain that is dark, so the dark green here or the dark gray, is constant. So if you drew a line here, as you go down, and on the homework it's a dotted line, everything below the dotted line is constant region. What that means is that within any isotype, it means the amino acid sequence is the same. So all IgG, why are they IgG? Because all IgG antibodies have the same amino acid sequence in their heavy chains and in their light chains in the constant region. IgA has a different amino acid sequence, and IgD has a different amino acid sequence. That's what puts them into these categories or isotypes. It has to do with the amino acid sequences in their constant regions. Above this dotted line here, the top of this, which is a lighter color of green or a lighter color of gray, that's referred to as the variable region. And the variable region, that's what allows antibodies to be antigen specific. So if you recall, I said there were many subpopulations of B cells and that they were actually genetically modified. So certain genes, it's a little bit like post-transcriptional modification. In post-transcriptional modification, there were sections called introns that were taken out and exons were put together. But this modification of B cells is DNA that's modified. Sections of genes that are going to code for antibody production are taken out and other sections are put together. So you get these different combinations. This allows the variable regions to be quite diverse. So we have in the IgG category, all the constant regions are the same, but the variable regions are different. And this is going to allow these antibodies to connect with different antigens. So they have specificity at their antigen binding site. So this is just repeating the same thing, same isotype, if they have the same amino acid sequence in their constant region, but their variable regions can be different, and it is dependent upon these different subpopulations of B cells. This is showing a diagrammatic representation of the antibody and kind of showing you how the variable region, the top, which is the antigen binding site, is very specific for a particular antigen. Here the antigens are shown with different colors and also different shapes, and they're showing that this specific antigen can bind with this specific variable region. It's handy that it's color-coded and also that it's shape dependent. But you can imagine there's lots and lots of different potential antigens, so you have to produce a wide variety of different antibodies that can bind with them. And again, the binding of antigen to antibody is not covalent bonding, it's either hydrogen bonding or ionic bonding. It's not permanent. So what are antigens then? Again, the term means antibody generator. These were thought to be foreign agents, and in some references they will say they are dangerous, something the body recognizes as being dangerous. That's a little bit hard to pin down, and sometimes these things are not foreign, but typically, again, when we're talking microbiology, antigens are bacteria. When I talk about foreign agents, I'm talking bacteria, fungi, protozoa, multicellular parasites, etc. But they could also be pollen grains, or they could be some type of large molecule. They do have to be pretty good size, and we'll see why in just a bit. So they can be cellular pathogens, but they can also be viruses, or they can be large molecules that are really not organisms specifically. They can't be too small. These things have on their surfaces a number of what are called chemically defined sites. They're called epitopes. They're also called antigenic determinant groups. So if you have a cellular antigen, especially if it's a big cell, say a trypanosome is going to have a very large variety of these antigenic determinant sites on its surface. Those individual sites are what bind to the antibodies. And so you can have big cellular antigens that will stimulate the production of many different antibodies. They're going to stimulate an increase in the populations of B cells that produce those antibodies. Here you can see the antigenic determinant group shown as kind of red spots on the surface. If I use this virus model, I'm going to be talking about virus in a little bit. If this were a measles virus, and we say, okay, this measles virus has a capsid, and here we see the capsid with its triangular shaped capsomeres, but we can look at this and we can say, clearly you've got at least two different types of protein, right? You've got yellow ones, and you've got green ones. These would represent antigenic determinants. Yellow antigenic determinants, green antigenic determinants. That's way more simplified because it's more complex. But you have to have at least two in order to stimulate an immune response. So to be an effective antigen, you have to have at least two epitopes, probably because all of the antibodies can bind at least two. Some can bind more than that. But you have to have at least two If you have less than that, then you have what's called a hapten. Hapten, the term hapten means incomplete antigen. A hapten can bind to an antibody if the antibody is already produced, but it will not stimulate antibody production. This is why sometimes very small molecules, if somebody wants to get an individual to produce antibodies against a very small molecule, they have to conjugate. They have to hook that molecule to something else. So they've got at least 2 antigenic determinant groups or 2 epitopes. I had my sister-in-law tell me at one point that her husband was allergic to salt. And my thought was, I don't think so. Salt is sodium chloride. That's way too small. There's no possibility that you can stimulate production of antibodies against salt. Now, salt may be in combination with something else. I don't know, but not salt by itself. Well, anyway. Now, antibodies can also be, antigens can be divided into categories based on where they come from. So if they are microorganisms that are coming from the external environment, again, bacteria, fungi, viruses, etc., they're called exogenous antigens. What that means is there must also be endogenous antigens. And these would be antigens that are part of yourself. Endogenous antigens are chemicals associated with your body. Ideally, as your immune system develops, you do not want to maintain populations of cells that will produce antibodies against endogenous antigens. You don't want to be producing an antibody against self. That's self-destructive. You got autoimmune disease going on. But we have lots of chemicals within our body that we could potentially produce antibodies against. So now this is going to show you kind of diagrammatically one way that a humoral immune response could be initiated. And what we're going to see is that this humoral immune response involves B cells, yes, because they produce antibodies, but it's also going to involve T cells. So it's going to involve the cellular immune response. It's also going to involve macrophages, phagocytic white blood cells. So it's going to involve innate immunity. So we get overlap between these different systems, which is not too surprising. Again, when you start looking at physiology, it tends to be very complex. So we've got B cells involved, we've got T cells involved, and we've got that thing which is supposed to be a monocyte. And that's a neutrophil. We can get neutrophils involved too sometimes. So we're going to be looking specifically at a T cell dependent mode of initiating a humoral immune response. Humoral immune response doesn't necessarily involve T cells and phagocytes, but we're going to look at one that does. So a pathogen comes in. We'll use this as a model. Pretend this is a measles virus. And in actuality, a measles virus is not a really good representation for something that would initiate a humoral immune response, because mostly they initiated a cellular immune response. But pretend that's not true. So here comes this virus. It gets in. To get in, that means it's gotten past our first line of defense. And what was our first line of defense? Skin and mucous membranes. Don't leave out mucous membranes. Skin is only on the dry surfaces of the body, but you have mucous membranes lining the GI tract, lining the respiratory system, lining the urogenital system, etc. Don't forget about the moist surfaces, because they're very, very important. Anyway, yeah, it has to get past that, skin and mucous membranes. So it's going to get into the circulation. So here we're looking at the inside of a blood vessel. You see some red blood cells coming along there, and you see these measles particles. They've gotten into the bloodstream. What's going to happen next? What's our second line of defense? Assuming we've got normal function. Something foreign gets in. Neutrophils and macrophages. Some white blood cells, some phagocytic white blood cells going to say, hey, what's that thing floating in here? And it's going to grab it and eat it. In these diagrammatic representations, again, you have to pretend that that thing is a measles virus on the far left. And you see a phagocytic cell kind of gripping onto the measles virus in the middle. And I'm not sure what's going on over there on the right-hand side, but that's supposed to be a white blood cell consuming the measles virus. So they're going to get taken in. They're taken in, and after they get taken into a white blood cell, they're brought in contact with the lysosome enzyme and the hydrolases, and they're digested. So the parts of the virus are taken apart. This virus is trying to fall apart already, so you can imagine it'd be pretty easy to take apart green parts and yellow parts that would be our antigenic determinant groups or epitopes. Then an interesting thing happens. There are quite a number of cells within our body that are referred to as antigen-presenting cells. And macrophages are like that, and neutrophils are like that. So after they have digested the virus, they're going to present the virus epitopes on the membrane surface. So the epitopes here are represented by green and yellow balls. They should have been triangles, but I'm not sure why. Maybe it was easier to draw balls, but anyway. So we've got green epitopes and we've got yellow epitopes, and they are in combination with these proteins that are associated with the macrophage surface. These proteins are referred to as major histocompatibility complex proteins. Anybody familiar with histocompatibility? Histo means what? Tissue. Is there a reason why we want to know about compatibility between tissue? If you've ever received a transplant, then you know, yes, it's very important to know. Are you going to be compatible? If you're receiving a tissue transplant, is there compatibility between your tissue and somebody else's? So these proteins, these major histocompatibility complex proteins, are associated with all eukaryotic cells. And they allow my cells to be recognized by me as self, and somebody else's cells to be recognized as not me, not self, or foreign. This is significant. So here's our phagocytic white blood cell. It's presenting mesal epitopes on the surface in combination with major histocompatibility complex proteins, or these are sometimes referred to as MHC protein. Again, MHC is major histocompatibility complex. Those things are also sometimes called human leukocyte antigens, or HLA. Sometimes, but not by me. I typically call them MHC proteins. Anyway, so then what happens? This is where our phagocyte is now presenting epitope in combination with MHC protein, and that's going to attract the attention of the next player in this story, and that's the T helper cell, or helper T cell. This is a T lymphocyte that has the CD4 marker on its membrane surface. It's going to recognize that viral epitope, and an interesting thing about T cells is they can only recognize antigen in combination with MHC protein. This influences what T cells can act against, which we'll talk a little bit more about later. But they recognize that macrophage and the MHC protein and the mesal epitope, and they are going to be stimulated to release some type of cytokine. At the same time, we have B lymphocytes. Again, B lymphocytes have antibody on their surface. If the antibody is the right type to interact with one of those mesal antigens, then the B cell will also bind. So you've got your macrophage with mesal epitopes on the surface. The T cell comes in, recognizes the epitope with MHC protein. The B cell comes in and recognizes the antigen. So they're right next to one another. And then the T cell releases interleukin. There are quite a number of different interleukins, and they have different numbers associated with them, and never mind. It's like there's way more detail than we need to get into for this class. Just know that T cells, helper T cells, release interleukin, and the function of interleukin is to cause cell proliferation. So what's going to happen is it's going to cause the B cells that are capable of binding to that specific mesal epitope to reproduce. You're going to get a whole bunch of B cells. And those B cells are then going to give rise to plasma cells. The plasma cells give rise to antibody in large quantity. At the same time, there are some B cells that stay as B memory cells. So in this diagram, here's our macrophage. You see the macrophage taking in the mesal virus. It's presenting these antigenic determinants or epitopes on the surface. Helper T cell recognizes that. Helper T cell then stimulates this B cell that also has antibodies against that mesal virus to reproduce, form clones. A clone is a whole bunch of cells of the same type. So you get a very large number of B cells. Most of those are going to be plasma cells that give rise to secreted antibody, protein that's going to go into the bloodstream. And then some of them become B memory cells that can persist within the body for years, sometimes the life of the individual. Well, so this is going to cause antibody titer to go up. This is about the point where I need to tell you guys a joke. Do you know why anteaters never get sick?
Why?
Because they're full of antibodies. Well, so you have an idea that antibodies are protective, right? They do something good for you. They protect you. But oftentimes we don't think about exactly how they do that. So the antibody titer is going to go up. That means you're going to get a very large quantity of antibody produced. And antibody titer is not really a measure of concentration. It's a measure of activity. There are a number of things that are measured by titer. Enzymes. We know what enzymes do. They catalyze reactions. So enzyme, you can talk about enzyme concentration, but you can also talk about enzyme titer and also antibody titer. It's a measure of activity. So that's going to go up. If the same pathogen, the same measles virus, gets into the body a second or subsequent time, oftentimes the B memory cells will trigger very rapid cloning. More B cells are going to be produced, and then you get a very large amount of antibody that will stay in the circulation for a long time. This is called an anamnestic response. This word, anamnestic, if you take it apart, we know that a or an in front of a word means without. So take the an off, and what you have left is amnesia. And what does amnesia mean? It means memory loss, or to forget. And you can't recall. And so anamnesis means without amnesia, and it means to recall. The body recalls seeing that antigen before, or encountering that antigen before, and it's going to produce a very large number of B cells and produce a whole bunch of antibody very quickly, very quickly. So in this diagram, what you see is, say the first time a person is exposed to the measles virus, they get a little bit of IgM and a little bit of IgG, but the levels go off. And then this is broken because you don't know how long a period of time has been passed, but at some point, that same measles virus gets in, and we get an anamnestic response. And IgM is about the same amount, but the IgG goes way up, and this is going to stay high for a very long period of time. Which, by the way, is the whole point of immunization, trying to protect somebody, stimulate antibody production. Well, okay, so this is showing the same kind of thing. Each subsequent exposure to the antigen, the number of different responding B cell clones will increase. If you had this, you're putting the same antigen in over and over again, you're going to stimulate more B cell memory cells to produce more plasma cells, and you get more and more antibody being released. And then the antibody is going to help protect you against that antigen. So again, in this particular example, we had our innate cells, the macrophages that consume the antigen in the first place, and then we had our cellular immune response cells, the T cells, the helper T cells, and then we had the B cells. All three are involved, both innate immunity and adaptive or acquired immunity, and both branches. And again, although the virus, the antigen used as our example is a measles virus, in actuality, measles virus tend to trigger a cellular immune response, just T cells, not antibody production. But my diagrams with the yellow and green kind of went along with our model. And I think this might have been a model of a measles virus. I'm not sure. We always call it that. It's like, I don't know. Anyway. Makes for a good story. Okay, so again, the memory function, this is why we say that this is the adaptive immune response, because you have this memory function. And if the antigen gets into the body a second or subsequent time, you're going to respond faster and with a more powerful response. You're going to get a lot more antibody produced very quickly. Now, so what? The question is, so what do antibodies do for us? As I say, anteaters don't get sick because they're full of antibodies. Well, what do antibodies do? So people have studied antibody-antigen interactions in vitro. These are called serological reactions. Serology is the study of antigen-antibody interactions in vitro. But in vitro is in an artificial container, so what happens inside the body? Well, we can kind of surmise what goes on inside the body by looking at these serological responses. and think about what kinds of things do we see going on inside the body. In tonight's laboratory, we're going to be doing diagnostic immunology, and we'll talk about 2 of these serological reactions. Again, the term applies to serum. Serum is blood plasma that has had the clotting factors removed, has a high antibody titer. So serological reactions, again, this is how antibodies interact with antigens in vitro, but they can have application within the body. The first type of reaction is called a precipitation reaction. Precipitation occurs when antibody and antigen bind, and the antigen is initially soluble, but when antibody binds to it, it makes it insoluble and it falls out of solution. It becomes a precipitate. And so what we see is this falling out of solution on this side. If this were in a tube, you could actually see this happening, but it does require a large quantity of antibody and antigen binding in order for that to be visible. Sometimes precipitation reactions are observed in tubes. Tonight we're going to be seeing a precipitation reaction occurring in agar. So we're going to run an Auchterlonie test, which is a form of precipitation reaction. And this is showing a real Auchterlonie test, and possibly our fake Auchterlonie test, which is what we do in the lab. Anyway, precipitation can occur in agar where antigen and antibody diffuse toward one another, and where they meet, they form the precipitate band, which in this case you can see is white. We're going to be doing Auchterlonie tests in lab. Remember to keep the slides that have the Auchterlonie test running on them over the dark bench. Don't put them on white paper because the precipitate's white. If you put the slide on the white surface, you can't see it. The next type of serological reaction is called agglutination. Now you've been introduced to the term hemagglutinin, remember? Who produced hemagglutinins? That you had to know. It was the influenza viruses. The influenza viruses had hemagglutinin on their surface. Hemagglutinins were proteins that were on the surface of the influenza virus. Well, agglutination means clumping. Hemagglutinin means clumping of red blood cells. And the influenza viruses cause that, but they're not the only thing that does that. So agglutination reactions occur when antibodies bind with cellular antigens and cause those antigens to stick together. They clump. This can be made visible. We're going to be doing hemagglutination tonight. What you see in the picture on the left is a blood smear, actually 3 little blood smears, where serum has been added, and if an agglutination reaction occurs, you see clumping on the surface of the slide. So you get to do hemagglutination. We're going to do blood typing tonight. You don't get to use your own blood. Sierra gave up letting students use their own blood quite a long time ago. Had to do with the price of serum going way up, and it had to do with HIV. We just don't do that anymore. So we're using completely fake material for our agglutination reaction, but it works pretty well. And you can see the same thing in the image that's on the right. Agglutination is not necessarily hemagglutination though, because agglutination can also be used to identify bacteria. So if you have a bacterial smear in liquid, you've all made smears on slides, so if you put some cells in liquid, and they're just kind of a uniform surface, if you mix antibody with them and the antibody sticks to them and causes them to clump, you'll get this separation into clumps, so it begins to look more like grated cheese or something in the liquid. Anyway, so that's an agglutination reaction. So when we talk about bacteria being identified relative to serological typing, that's what it is. It's agglutination reactions. And salmonella, there are a whole bunch of different salmonella serotypes. This is showing direct agglutination on a slide. So if you have cells in liquid, this is just a uniform mixture. But if you have cells stuck together by antibody, you get this kind of look. There's a lot of application for this type of thing in diagnostics. Another type of serological reaction is called neutralization. Neutralization, that means you're going to make a chemical no longer active, and what typically gets neutralized relative to bacterial pathogens is toxins that they produce. A number of bacteria cause disease by producing protein exotoxins. And if antibodies bind to those toxins and neutralize them, then the toxins can't cause harm anymore. We produce antibodies against tetanus toxin, which is why you want to have your tetanus vaccine kept up to date, because what you're given is a toxoid. It's a detoxified microbial toxin. It can't harm you, but it will stimulate antibody production. You want to make sure that you maintain fairly high levels of antibody against the tetanus toxin, because the amount of tetanus toxin required to kill you is significantly less than the amount of tetanus toxin required to initiate a humoral immune response. If you mess around with that one and wait until you're exposed to the real toxin, it's probably going to kill you. That's not fun. Okay, another reaction is immobilization, but this doesn't necessarily mean antibody binding to flagella or even pili. It means that antibodies are going to cover up the binding sites on cells, pathogens that would try to attach to host cells. The pathogens could be viruses, they could be bacteria, they could be protozoa. Antibodies stick to them and cover up their binding sites. If the pathogens can't bind to the host, they can't infect. So this is a very effective means of blocking their entry into cells. And again, you can see viruses there with antibodies stuck to them. The viruses then can't invade the red blood cells. Another serological reaction is opsonization. And I know I mentioned opsonization earlier. Opsonization means that antibodies are going to stick to the surface of the antigen and make that antigen more attracted to phagocytes. It's somewhat complex because the antibodies, if you look at that little Y shape, the top of the Y is where those antigen binding sites are. But the bottom of the Y, if you looked at, this is an antibody, so down here about elbow, there's a region of the antibody that's called the Fc fragment. And the Fc fragment is very attractive to phagocytes. If antibodies are stuck on the surface of a cell, what you have are all these Fc fragments sticking out, and the phagocytes look at that and recognize that it's something they want to attach to. So having antibodies stuck to the surface of something makes them attractive to phagocytes. This is opsonization. But if you'll notice in this diagram, antibodies are not the only things causing opsonization. The other thing that I mentioned that causes opsonization is complement. And the complement are those little gray balls that are on there. And then the other thing that antibodies binding antigens can do is they can trigger the complement cascade. The complement proteins C1 through C9, typically starting with C3 that splits in half in C3a and C3b, and then you get all these other complement proteins coming into play. A cascade effect that ends up with the membrane attack complex, which is C7, 8, 9, and the 9 makes holes in cell membranes, and then those cells blow up. If you make a hole in the membrane that allows water to come in, it will, because the protoplasm has a higher solute content, water will go in and the cell will blow up, unless it has a wall. So, again, we see that antibodies can sometimes control a pathogen. If we're talking about a protein exotoxin produced by a pathogen, they can neutralize that. The antibodies can prevent the pathogen from binding. They can immobilize them. But most of the other ways that antibodies work involve these other cells. They can get the phagocytic cells to get involved in there, or they can bring complement factors in, or sometimes they will trigger the activity of NK cells, the natural killer cells. Well, okay, so our body has the ability to produce antibodies. People vary quite a bit in terms of their genetics. Again, if we're talking about a normal, healthy individual, you can produce different antibodies that can specifically bind to a myriad of different antigens, probably thousands of them. The question is, which kinds of things should you produce antibodies against? And as I said, you don't want to produce antibodies against self, and you also want to be not necessarily producing antibodies against everything that's foreign, because we encounter quite a lot of foreign substances in the air, in our food, etc. So how do we not respond to every foreign substance? And the answer is immune tolerance. So immune tolerance, this is our ability to not mount an immune response against certain types of foreign substances. Or, as I said, you don't want to mount an immune response against self. So immune tolerance allows you not to. If we look at what happens, actually what allows us to have immune tolerance, we have mechanisms that eliminate cells that would produce antibodies against self. We also eliminate most of our T cells, that is T cells that would attack our own cells. So if you remember, and I talked about that a little bit before, you have many, many, many subpopulations, but a lot of them get taken out of the circulation, or they are eliminated completely. Those cells are killed off. But again, we're exposed to pollen grains, fungal spores, lots of different chemicals in food. Now we have all kinds of things that are sprayed into the environment around us. And anybody know what that image on the bottom right corner is? Those are sperm cells. So it'd be pretty tough for a woman to get pregnant if she was mounting an immune response against spermatozoa. They're foreign, they're cells. Sometimes people do produce antibodies against those, and then pregnancy is really going to be difficult. But most of the time we don't. And it's because of immune tolerance. So, again, we normally do not produce antibodies or immune cells against self. Cells that would have given rise to antibodies against self are taken out of the population. It's called clonal selection. So we remove certain types of cells. This is going to help to prevent autoimmune disorder, where we mount an immune response against our own cells or our own tissues. That's not a good thing. So for a long time, people weren't sure exactly how this happened. Now, as I said, they're pretty certain that what happens is your body produces a very large array of subpopulations of cells, and most of them, the ones that would mount an immune response against your own tissues, your own molecules, they're removed so that they cannot later on give rise to antibodies or T cells against you. And both B cells and T cells are subject to this. So we've just talked about humoral immunity, and again, humoral immunity involves B cells and the production of antibodies. You want to be able to produce antibodies against foreign agents. You do not want to produce antibodies against your own cells. If you did, again, you would damage your own cells and tissues. Now, we know that microorganisms that live in the gut have antigenic determinants very similar to certain types of human cells. And so we're going to talk about blood typing, where people that have O-type blood will typically produce antibodies against both A antigens and B antigens that are associated with some human blood cells, even though you've never been exposed to them. And the question was always, why? Why would you produce antibodies against something that you've never been exposed to? It turns out because we have bacteria living in our gut that have antigenic determinant sites very similar to the A antigens and B antigens. So we've been exposed to those antigens because of the bacteria living in our gut. And people now think that that's one of the roles that the normal flora play, is that they help to get your immune system functioning properly so that you don't necessarily produce antibodies against certain kinds of things, and you do produce antibodies against other kinds of things. This is just a statement that children that are raised in extremely clean environments, where they're not exposed to a lot of bacteria, often have a lot more trouble with allergies. They tend to have hypersensitivity to a lot of foreign agents, and it's because their immune system is not properly primed by bacteria, which it would have been if they were living in a grubbier situation. So if your children want to go out and play in the dirt, let them play in the dirt. They want to pick up rocks out of the driveway and put them in their mouth, it's yeah, you go for it. Because that's what they used to do. And that's how you get your normal microbiota established. Okay, so that was humoral immunity. Now we're going to talk about cell-mediated or cellular immunity. Again, this involves T cells, T lymphocytes, and cytokines, because T lymphocytes don't produce antibodies. There's a lot of diagrams in here. Most of these are coming off. Actually, you just Google something and then ask for images, and you get all kinds of weird pictures. So T cells, as I said, do not produce antibodies. They also respond a little bit differently than B cells, because T cells can't respond to any antigen that comes along, they can only respond to antigens in combination with MHC proteins. So there's something about the receptors on their surface. They have to see the MHC protein and the antigen together. Otherwise, they can't respond to it. This means T cells have only specific targets. Again, MHC proteins are on eukaryotic cells. So T cells cannot respond to free virus particles because they're not cellular. And they cannot respond to free bacteria because bacteria are prokaryotic and don't have their own MHC proteins. They can respond to viruses or bacteria stuck to host cells. So if a virus binds to a host cell, then you've got virus plus MHC protein. Or if a bacterium is stuck to a host cell, then you've got bacterium plus MHC protein. But those guys alone, T cells cannot respond. So T lymphocytes, then if we think about what kinds of antigens can they go after, they can go after anything eukaryotic. So they can go after fungi, protozoa, multicellular parasites. They will also recognize tumor cells. Tumor cells are eukaryotic and they're a little bit armed. They start expressing weird membrane markers. They can go after infected body cells, and they can go after transplanted tissues. If you receive tissue from an external source and the major histocompatibility proteins aren't compatible, so they're not matching you, then they're foreign and the T cells can attack. So they can, like I said, kill infected body cells. The T cells that are the fighting arm of the cell-mediated immune response are called cytotoxic T cells, which is kind of a misnomer because they're not really producing toxic substances, but they do produce granzymes and perforin. Perforin makes holes in cells, and granzymes destroy proteins. So if killer T cells go after a cell that's been infected by viruses, they'll not only kill the cell, they'll destroy the viruses too. They'll take them out. So again, there are multiple subpopulations of T cells. We said that cells that leave the red bone marrow, if they're going to become T cells, they have to migrate into the thymus gland, which tells you a little bit about cell-mediated immunity. It's very robust in young individuals because the thymus gland is quite large. As individuals age, their thymus shrinks until it almost completely disappears. So older individuals begin to lose their ability to produce T cells. They have to go to the thymus gland for processing. Remember, the thymus gland produces thymosin and thymopoietin. These hormones are essential for T cell development. So then you get all these different subpopulations of T cells, and they are categorized in these two major groups by their membrane markers. You've got CD4s and CD8. But they're carrying CD4 markers, we now call them helper T cells. You used to call some helpers and some inducers, and at some point they stopped that division and just said if they've got CD4 markers, they're all helpers. T helpers or helper Ts. So these T helper cells, they have a whole variety of functions, and we just saw them helping to initiate a humoral immune response. So they've got CD4 membrane markers. There are not just one type of CD4, there are subpopulations. So there's many different subpopulations of these helper T cells, and they can produce a variety of different cytokines that have multiple different functions. So one of the things that we saw our helper T cells doing, as I said, was releasing interleukin that caused proliferation of B cells. So they play a major role in getting a humoral immune response started. But again, that's not all they do. So interleukin, and as I said, there's many different types of interleukins. Don't worry about the numbers. There's like interleukin 1, 2, 3, 4, 5. Never mind. We're just not going to go there. But interleukin is something that causes cells to proliferate. It was necessary to get clones of B cells. It's also necessary to get more T cells. Another thing that helper T cells release is a substance called gamma interferon, or interferon gamma. Gamma interferon is a little bit, it's not, so I talk about interferons being chemicals that interfere with cytolytic virus life cycles. That's what they're named for. But gamma interferon acts on phagocytes. It apparently attracts phagocytes to come to an area, and then it also activates them, so they run around and consume things rapidly. So it brings in the innate immune response, getting the phagocytes involved. Helper T cells can also produce a substance called tumor necrosis factor. And this is one of the problems with cytokines. When you look in immunology textbooks, you'll see a lot of these things have multiple different names. I'm not sure why a lot of chemicals are like that. Even enzymes are like that. So tumor necrosis factor is also called lymphotoxin, and it causes tumors to die. It interferes with their the circulation, so it removes their blood supply. They can't get nutrients, and they die. This is talking about the details of T cell processing. So after they leave the thymus gland, or what happens to them in the thymus gland, never mind. So, no, we're not going there. You could spend a lot of time talking about this. So they're going to be involved in initiating that humoral immune response, again, by releasing interleukins and getting the B cells to reproduce. They're also going to influence the reproduction and differentiation of other T cells, which is essential to the cellular immune response. And then this is talking about HIV. The primary target for HIV, human immunodeficiency virus, is helper T cells. So what does HIV do? It kills off your T4 lymphocytes. Without your T4 lymphocytes, you can't stimulate cell proliferation, which means you can't produce clones of B cells, which means you're not going to be able to produce enough antibody to react against a foreign agent that gets in. Basically, the humoral immune response begins to fail. But helper T cells also stimulate the reproduction of other T cells. So your cell-mediated immune response also fails. Without your immune response, then you're susceptible to infection with anything that comes along. And typically people that have infection with HIV, supposedly it can take up to 20 years for symptoms to begin to develop, but eventually their immune system fails and they begin to be infected by other kinds of things, and they will die from tuberculosis or some other type of infection because their immune system fails. And so the associated syndrome, which is acquired immune deficiency syndrome, some people on, I think it was the last quiz, where I said, what's the name of the virus? The name of the virus is HIV. AIDS is not a virus. AIDS is a disease syndrome, acquired immune deficiency syndrome. It's a disease. Okay, the other type of T cells I want to talk about are CD8s. So these have the CD8 markers on their membranes. One category of CD8s are called killer T cells or cytotoxic T cells. And again, these are the primary defending arm of the immune system, the cellular immune system. So these guys release perforin and granzyme. Perforin makes holes in cell membranes, and granzymes destroy proteins within cells that they attack. So if the killer T cells go after cells, and again, they can only go after eukaryotic pathogens, or they can go after infected cells, either viral infected cells or bacterial infected cells. They're going to kill those cells. They're going to make holes in them, blow them up, and they're going to destroy proteins within them. So again, eukaryotic pathogens, infected body cells, tumor cells, but they do not kill antigen-presenting macrophages. So this is kind of an interesting twist. We have these cells, these killer T lymphocytes, that can recognize antigen in combination with MHC protein. They can kill any kind of eukaryotic pathogen. They can kill infected body cells, tumor cells, transplanted tissue. Why do they not kill macrophages that are presenting antigenic determinant groups in the first place? Anybody know? Well, it turns out the reason is because there's more than one type of MHC protein, those major histocompatibility proteins. So our blood cells, not just the T cells, but all kinds of blood cells, the killer T cells recognize MHC class 1, and the blood cells carry MHC class 2. So your macrophages carry MHC class 2, also your red blood cells. It would not be good if your cytotoxic T cells killed red blood cells that were transfused, because their MHC proteins didn't match. So they don't, because blood cells, all blood cells, have MHC class 2 proteins, and T cells only go after MHC class 1. This is what allows blood transfusions to even be possible. That's not taking into account the other antigenic groups that are on blood cells, which we'll talk tonight, and we'll talk in lab about those problems. The other type of CD8 cells are called regulatory T cells. And in the notes, they still may be called suppressor T cells. Apparently the term suppressor has been eliminated, and now these are called T regulatory cells or regulatory T cells. So if you see them referred to as T regs, that's T regulatory cells. They still have the same function, and that is to shut down the immune response. So if you want to think about them as suppressing the immune response, that's fine. They were initially referred to as suppressor T cells, and their function is to turn off the immune response sometime after it's been initiated. The reason that we need this to happen is because there can be an excessive immune response. We're going to get into this. It's called hypersensitivity, where an immune response can actually begin to do tissue damage, damaging your own cells and tissues, and that's not okay. So the Tregs, their job is to turn the immune response off and try to prevent hypersensitivity. This is just a little note that says, yes, the immune system is very complex, way more complex than what you're getting here. You can take an entire course in immunology. So you can imagine, it's like you can take an entire course in virology. What you're getting here is just brushing the surface. Always. This is an introductory class. Okay, so now, having at least some idea, that is not what I wanted to do, get back there. Having some idea how the immune system works, now we want to talk about immunization and hypersensitivity. We'll see how far we get with this. So, a lot of what I'm presenting now, at least, is somewhat familiar. You have an idea of what antibodies are. You have an idea about what immunization is, but it might not be exactly what you think. So when we're talking about the history of microbiology, I mentioned that it was Louis Pasteur who discovered that attenuated microbial cultures could be used to prevent disease. And Pasteur used the term vaccine to describe these attenuated microbial cultures. He was giving credit to Edward Jenner. Jenner had been immunizing people using the fluid from cowpox blisters or cowpox pustules. So here we see Pasteur doing something, I'm not sure, but anyway, and then you see some microbes. So microorganisms, if they're attenuated, they're weakened in some way, they can often stimulate an immune response but not cause disease. And it was Pasteur that found that out kind of accidentally. But we know about other successful studies. Pasteur, Robert Koch, Edward Jenner, Joseph Lister. A lot of these guys were doing kind of investigations that involved the immune system kind of tangentially. They didn't really understand the immune system the way we understand it now. In fact, if you go back and look at a study of the immune system, what you find out is people really began to understand how the immune system worked when it didn't. So it was in the 80s, mostly in the mid and later 80s, that people really began to get a handle on how the immune system worked, because it didn't work, and it was because HIV was wiping it out. And that's when they found out, oh, guess what? If you take out the T4 lymphocytes, nothing works. So then they could kind of take it apart and work backward and understand how the system worked. Well, so these people were really important to microbiology, but if you look at what they knew versus what you know, or at least what I hope you know, that's how we get to the finals, they really did not understand a lot of these systems nearly as well. So now we're going to talk about immunization, and we're going to find out that what makes immunization works is the anamnestic response. This rapid increase in antibody titer, if you're talking humoral immunity, it's rapid increase in antibody titer following a second or subsequent exposure to the same antigen. So remember what anamnesis means to recall. What is immunization then? Immunization is the process of conferring specific immunity by artificial means. Now think back when I started talking about adaptive or acquired immunity. And I said adaptive or acquired immunity could be acquired either naturally or artificially. If it's natural acquisition, it could have been through get the disease, hopefully survive, recover, be immune. Or you could naturally acquire immunity through a mother's breast milk or through the placenta. But now we're talking about artificial means. And remember that artificial can be either active or passive, so we have to look at both. But immunization is this process of conferring specific immunity by artificial means. If it's going to be active, then we want to get the body to produce its own immune cells and substances. But it could be passive, where you just give immune cells or substances to the person rather than inducing them to produce their own. So immunization can involve vaccination, and unfortunately those terms are often used interchangeably. They do not mean the same thing, because immunization is just it's conferring specific immunity by artificial means. It can be either active or passive. Vaccination is active. Vaccination is inducing active immunity. And you're inducing active immunity by putting something into the body that says to the body, you immune cells, get going. You B cells, reproduce, form clones. Clones are going to make antibodies. Or you T cells, reproduce, make more T cells. So if a person is vaccinated, they are going to be induced to respond. Their body's going to produce its own immune cells and substances. And again, the term vaccine was applied by Louis Pasteur. He was giving credit to Edward Jenner. So a vaccine is a substance containing killed or attenuated microorganisms. Initially, it was killed or attenuated microorganisms. Currently, we have other ways to generate vaccines because we have a lot of methods that involve recombinant DNA techniques and so forth, and a lot of manipulation of molecules. So sometimes our vaccines are not whole cells. They can be parts of cells, but they can also be substances associated with cells. So oftentimes you'll have what are called subunit vaccines, where there's some component, an epitope, if you will, that's some part of a cell, but it's not the whole thing. So it's not necessarily killed or attenuated microorganisms anymore. And then I mentioned that sometimes disease is caused not by cells, but by substances cells produce. So for example, if you get strep throat, the organisms that cause strep throat are living in your pharynx, but they're producing hemolysins, toxins that go into your bloodstream, and that's what's causing damage. If you get diphtheria, the Corynebacterium that causes diphtheria is living in your pharynx, but they're producing toxins that go into the bloodstream, and it's those toxins that cause disease symptoms. If you get tetanus, it's a toxin that causes tetanus. It's not Clostridium tetani. It's the toxins produced by them. So another way that you can be immunized against something is to receive what's called a toxoid. A toxoid is a detoxified microbial toxin. So it's the tetanus toxin or the diphtheria toxin or some other toxin. It's been modified so that it can no longer cause damage, but it will stimulate an immune response. Now, if you get what's called a combined vaccine, something like the DPT, or now it's called Tdap or DTaP, this is a combination vaccine and toxoid, and actually it's mostly toxoid. So this is diphtheria, tetanus, and pertussis. Both the diphtheria and the tetanus, those diseases are caused by toxin. So you're getting toxoid, detoxified microbial toxin, that you will produce antibodies against. And then the pertussis, that's actually a vaccine, where you're going to produce antibodies against the bacterium that causes whooping cough. And one of the things we talked about when talking about viruses and viruses getting into cells, viruses can sometimes put genes into the chromosome of a host cell that causes the cell to produce toxins. We now know that the tetanus toxin, tetanus toxin, botulism toxin, the toxin produced by Corynebacterium diphtheria, and quite a number of other toxins are the result of lysogenic conversion. So those toxins are actually encoded by viral genes. Well, okay. So vaccination, again, the act of stimulating active immunity by artificial means can be done either using a vaccine or using a toxoid. In either case, this is only going to work if a person has the ability to mount an immune response. If the person is immunocompromised, they don't have T cells, they've lost their helper T cells, they're not going to be able to respond to this. It's not going to work. So then what can you do to immunize them? Immunization is still this process of conferring specific immunity by artificial means. if they can't produce their own immune substances, you can give them pre-made immune substances. So you can give them pre-made antibody. This is called anti-serum. In this case, it's artificial and passive. You're giving a person something that is already made for them. This used to be done typically by inoculating some kind of other animal and getting that animal to produce antibodies. Most commonly, the antibody that is administered is IgG, or gamma globulin. It is the most important antibody that's in our defense. And again, what people used to do is inoculate some large kind of animal, oftentimes a horse, sometimes goats, cattle, but they would immunize those guys, and then they would produce antibodies, and then they would take the horse antibodies and give those to people. That had serious drawbacks because they weren't human antibodies. They were horse antibodies. And the first time you do it, maybe you're okay, but what's going to happen is your body's going to recognize horse antibodies as foreign, and if they get into your body a second or subsequent time, you're going to produce antibodies against those. We can produce what are called anti-isotype antibodies. Those are antibodies against antibodies. And then you have all kinds of problems. So they don't typically use horses to produce antibody anymore. Instead, we produce what are called monoclonal antibodies. So there are cells, basically cancer cells, that can be modified to produce very specific antibodies. And these cancer cells are immortal. They'll just keep growing. They grow in a tissue culture. They just live forever, and they can stimulate them to produce specific types of antibody, and it's human antibody. You don't have to worry about the anti-isotype antibodies being produced. So that kind of tells us what is immunization then. Again, it's conferring specific immunity. It can either be active, using a vaccine or a toxoid, or it can be passive, using immune serum. So under what circumstances do we use immunization? Immunization is considered appropriate for the mass of the population, which basically means everybody in your population, if two circumstances exist. One, if the disease that you're trying to protect people against is one that's going to spread rapidly, so it's going to move through the population and infect a very large number of people. That's one of the things you have to take into account. The second thing you take into account, well, this is showing you how disease can be spread. Again, droplet nuclei being released. The second factor is that the risk of contracting the disease far outweighs the risk of being immunized. Consequently, there are certain kinds of diseases that spread rapidly, but we do not immunize against. One of them is rhinovirus, the common cold. And you're unlikely to see a vaccine developed against rhinoviruses for two reasons. One, there's so many different kinds. There's like 100 different strains of them, so you'd have to have so many different vaccines. But the bigger reason is because the risk isn't that great. You don't die from having a cold. You get sick and you feel miserable, but typically it's gone in a couple of weeks, and then you get better and you're fine. So we don't immunize against the common cold. So a disease has to be one that's likely to spread rapidly, and it has to be more damaging, greater potential risk than the risk of being immunized. But that says, there must be a risk associated with immunization. Not nearly as much as you might think listening to some people on the internet or reading what some people have to say on the internet. The risk from immunization is far less than getting in your car and driving on the freeway. And we do that every day without question because we know we have to. Actually, some people try to avoid it and go around on the back roads. Might be more dangerous, but anyway. So if we look at vaccine-preventable diseases, we can look at what kinds of things the CDC recommends people be immunized against. This is not something that I expect you to memorize. You can look these up. Just go to the CDC website and see what they say. So for children from birth to 6 years of age, they need to be immunized against chickenpox, diphtheria, the Haemophilus influenza type meningitis, hepatitis A, hepatitis B, influenza, measles, mumps, pertussis, polio, pneumococcal pneumonia, rotavirus, rubella, and tetanus. Now, some people that argue against immunizing children say, I don't want my children to be exposed to all of those things all at once. Why can't they be given in little tiny doses, and maybe a few now and a few later? And do you think when you take your child out into the real world, you get to make that choice? I only want my child exposed to certain antigens. today they can get exposed to A, B, and C. Tomorrow, D, E, and F. It's like if they're out there in the real world, guess what, people? The antigens are all around you. Yeah.
So I have to ask for on this, is there a correlation to it causing, like getting antigens, like is there a correlation to it causing, like, what they call autism?
No.
I think, okay.
No. That's been completely debunked. There's absolutely no evidence. The person that was doing that actually got banned from being a physician in the British Isles, which is where he was initially practicing, so he moved to Texas.
Oh, good.
So no, there isn't. There's none. But if you read about autism, there's a lot of information that happened that kind of went along with this idea that, oh, there's more autism now. Actually, that was because they changed how it's diagnosed. Yeah, there is a lot more recognition of the autism spectrum because they changed how it's diagnosed. But no, sorry. Anyway. Okay. Anyway, you can look these things up. I stuck these slides in here because you can see what kinds of disease. A lot of the diseases that people are immunized against, we have never encountered in this country because we're immunized against them. And so you don't think about things like whooping cough. When was the last time you ever met somebody that had whooping cough?
There was just an outbreak.
Yeah, and there have been, and this is because people are now choosing not to immunize. Yeah, and diphtheria too, unfortunately. Vaccine preventable diseases, age 7 to 18 years. Again, influenza, tetanus, diphtheria, pertussis, the human papillomavirus, HPV, which causes cervical cancer and other kinds of cancer, meningitis, hepatitis A, B, polio, measles, mumps, rubella, and chickenpox. So yeah, there's a bunch of things you're supposed to be immunized against in order to prevent people from getting those diseases. You can go to the CDC site and you can look at the history of immunization and you can look at the number of cases of disease and how many deaths were caused prior to the availability of immunization. And it must be very, very frustrating to be a part of the CDC and be trying to tell people you need to be immunized and have people go, no, I'm not going to. Because I heard it causes autism. I'm not going to. My kid's not going to get that. And yeah. It goes on and on. There's lots of these. Well, okay. Nevertheless, there have to be some risks, and I do need to talk about some of the risks potentially associated with immunization. One is vaccine failure. In this country, I don't think vaccine failure has been an issue since quite a number of years ago. There was a batch of pertussis vaccine that didn't work, and it had to do with the manufacturer doing something wrong. And then people got wind of that and it's like, oh, don't get immunized against that because it doesn't work. It didn't work one time. But anyway, this is more of a problem in other places because oftentimes vaccines, in order to be effective, have to be stored under just the right circumstances. They have to be maintained at cold temperatures. They have to be maintained only certain periods of time, that kind of thing. And oftentimes they're not. So yeah, there's a potential for vaccine failure. Probably not so much in this country, but in other places, definitely. And then you think you've been protected and you're not. Another potential risk is infection. Is it possible if you're using a killed or attenuated, not if it's killed, but if it's an attenuated microbe, could it potentially become an infectious agent? And apparently, yes, that is a risk, particularly certain types of viruses. If you're using live viral vaccines, sometimes those things can cross the placenta. They didn't cause a disease in the adult, but if the adult person is pregnant, they can cross the placenta and infect the fetus. So you're not supposed to use live virus vaccines if you're a pregnant woman. They might also be able to cause infection in immunocompromised people that are sharing your household. Apparently this has also happened. Another problem is contamination. Again, probably not a problem in this country because you use a syringe one time and then it's discarded. But there are many places in the world where syringes are used over and over again. And I don't remember if I showed you the same parasite film that I've often showed where they had these little bowls of some kind of liquid and they were using sharp-pointed sticks, dipping them into the liquid and poking people with it. And my thought was, oh my word. Ugh. The potential for contamination was extreme. So yes, contamination can be a problem. Another potential problem is toxicity. Certain types of, if you're using killed bacteria and they're gram-negative bacteria, remember that the gram-negative cell wall has lipopolysaccharide in it. Sometimes that lipopolysaccharide can cause a toxic response. You can get really sick from being immunized. And then there's a potential for hypersensitivity. Hypersensitivity is an abnormal immune response where the immune reaction causes tissue damage or malfunction. But notice the images here. Although immunization is one potential cause for hypersensitivity, bee venom is much more likely to be a cause. So from here, this goes into hypersensitivity, and I'm not going to do that because we have to stop. So this is just talking about individuals arguing against immunization. Again, way less dangerous than driving on the freeway. But, so next time we'll talk about hypersensitivity and herd immunity also. I don't have time to get into that. We'll have to finish this lecture next time. So, the question has come up, what happens next Tuesday? You guys have a quiz. The answer is yes, but it will be a take-home quiz. I will give you that quiz on Tuesday. But here's the thing. This is the last quiz for the rest of the semester. You have a homework assignment, the Adaptive and Acquired Immunity homework assignment, that pretty much covers humoral immunity, cell-mediated immunity, talks about serological reactions, etc., etc. So the quiz that I'm going to give you will be a quiz that is about immunization, hypersensitivity, and then the rest of the topics that we have to cover, which are epidemiology and mechanisms of pathogenicity. So it'll be the last part of the material. It will be a take-home. The hardest part about that quiz is getting it back to me. Don't forget to give it back. Okay.
Will this one also be out of 25, like the first, or like the other take-home quiz?
It could be. You want it to be out of 25? I don't care.
Yes.
Sure. The more the merrier. The more points, the better.
Okay. Good.
We like those heavy point value take-home quizzes. I could just pick anything that we've had, but we haven't been out.
Perfect.
I like that.
That works.
Deal.
Okay.
Cool.
Since it's take-home, it's fine.
Woo.
Chance to grade your physiological unknown 1 report and get it back to you so that you can use it to study for the lab exam, which is next Thursday. The only way I can do that is if you give it to me tonight so that I can grade it and give it back to you on Tuesday.
Cool.